Okay, I wanted to take some time and go over some of the light-dependent reactions since we've had a couple snow days and I don't want to get too far behind. So I'll talk about some things up to the light-dependent reactions and then talk about them, and then hopefully we can get questions on Monday about them, or not Monday, but later on the week after our lab, and then we can head into the Calvin cycle. All right, so the big question is, like we've been talking about, how do living systems process energy? And we've seen eukaryotes processing energy through cellular respiration, but there's a second way, and that way is through autotrophism, primarily photosynthesis. So you guys know that there is a reciprocal relationship between animals and plants. That relationship actually exists by itself in plants, but if we step away from plants and think about the ecosystem, then of course plants and non-plants who are eukaryotes have a sort of a trade-off. And as you see in the bottom portion of the frame, the two equations for photosynthesis and cellular respiration, very similar, just sort of flip-flopped. Over the arrow, in photosynthesis, we would have sunlight providing the energy, and anaerobic cellular respiration providing the energy is glucose. Okay. Also remember that we have a tendency to think about plants all the time because we're surrounded by them, but there's an equal amount of aquatic photosynthetic organisms than there are plants on land. In fact, some of those photosynthetic organisms are not plants or eukaryotes at all. They're bacteria. And they were actually, cyanobacteria were some of the first bacteria to generate oxygen in the atmosphere. Eventually, evolution provided algae, which are protozoans, and they live as colonies. And then eventually we get things like seaweed, which are large colonies of protists living together, not plants. So lots of photosynthetic autotrophs, and of course we're not going to talk about them a whole lot right now, but we have chemotrophs too, which eat chemicals and give off waste products from that. You usually find those at the bottom of the ocean, at hydrothermal vents from volcanoes. They're eating sulfur and they're eating the chemicals that the early atmosphere was made of, and that was primarily gases from volcanoes. As a refresher, if you remember plant anatomy, we have leaves, which are primarily doing photosynthesis, really anywhere there's chlorophyll. In the leaves, we have a specialized group of cells called the spongy mesophyll. So let me zoom in on that real quick. And so the spongy mesophyll is in here, and sandwiched in the spongy mesophyll is the vascular tissue of the plant. Here's a vein of the leaf. The stomata, whether they're open or closed, are the anatomical features responsible for exchanging gases across the leaf. And so if we go even deeper into one of these chloroplasts in the spongy mesophyll cell, we can see these stacks of things called thylakoids. And then when they're in a stack, they're called grana. The thylakoid is interesting because it's in a container called the chloroplast, a double membrane. And inside there's a space, and this space is called the stroma. And in the stroma are the thylakoids, the stacks of thylakoids. And they are what's green. That's where the chlorophyll is, in the membrane of the thylakoids. And then inside the thylakoid, it's almost like a hollowed out disc. We've got places for the light-dependent reactions to happen. So the way this works is that the Calvin cycle will happen in the stroma, which is a jelly-like substance. And across the thylakoid membrane is where you have the light-dependent reactions happening, which sort of happen first. All right, so we know that light is essential for photosynthesis, and light is a form of radiation coming from the sun. We get it about every 7 minutes. And of course, it's only a small piece of the radiation that's coming to Earth, and so the visible light, that is. So that's what we're really concerned with here. Plants are using a narrow range of the visible light spectrum because they contain two pigments called chlorophyll. And the chlorophyll A and chlorophyll B are very interesting. If you look at the way the chlorophyll looks, it's basically has a lipid hydrocarbon tail and this big top, almost like a phospholipid looking, called the porphyrin ring. And in the middle, there's this tiny little magnesium atom in the middle. And so it is one of the photosynthetic pigments, but not the only one. There are things called carotenoid pigments, and there are xanthophylls, which are also found, and there are all kinds of colors. Oranges, purples, things like that. And they absorb a wider range of light, and of course they help the chlorophyll not break down. And you can see these pigments in the fall when leaves change color. All right, so why are plants green? Well, most kids know that absorption and reflection is the reason why anything has color. So chloroplasts don't absorb all the wavelengths of light equally. So they're absorbing specific blues and reds on the visible spectrum, and they're reflecting green. And it's the least useful for photosynthesis, and they're reflecting that. And so you can sort of see the pigments in plants here. Carotenoid pigments are more broad in their range of absorption, but chlorophyll A and chlorophyll B have these nice little peaks, and they absorb specific wavelengths of light, which we'll talk about in the light-dependent reactions. All right, so let's look at the light and photosynthesis. So if you look at the way photosynthesis is in contrast to cellular respiration, photosynthesis is anabolic. Okay, we're building a molecule. What are we building? We're building, in this equation anyway, glucose. You'll find out that we're really not building glucose. We're building a molecule called G3P, which is glyceraldehyde 3-phosphate. But in the equation, most of the time, G3P is being put together into glucose. So we're building glucose and we're liberating oxygen. So this is the main thing we're building. And what are we building it out of? We're building it out of these products here. So carbon dioxide is reduced. So we're adding electrons to build glucose. And water is oxidized. So we're losing electrons and we're liberating oxygen. So photosynthesis is anabolic, and it's also endergonic. So if we were looking at the picture that I've shown you before about Gibbs free energy, we'd be basically pushing the molecules up the hill, putting energy in rather than releasing energy. Okay, well, let's step back here a second. All right, and we've already talked about the chloroplast, so I'm going to go ahead through this and go to the next slide. All right, so photosynthesis is a two-part process. The very first part are called the light-dependent reactions, and there are two photosystems involved here that absorb two different wavelengths of light. So the light-dependent reactions occur in the thylakoid membranes, very similar to the electron transport chain happening across the intermitochondrial membrane. It's really uncanny how similar the chloroplast and the mitochondria are, except for in reverse. So light is used as the energy source to push the reaction along. There's a new electron shuttle that we're going to find called NADP. And when NADP gets reduced, it gets reduced into NADPH. Very similar to what NAD and NADH are. Okay, so we have two things going on in the light-dependent reactions. We're basically producing NADPH, and a lot of kids forget, we also produce ATP in the light-dependent reactions, and we give off oxygen. Now, one thing to think about here, which we don't really have to know a whole lot anymore for the course because it's changed, unfortunately, is that the light-dependent reactions generate NADPH and ATP, but not equally. In fact, the Calvin cycle uses ATP and NADPH unequally, and we'll get to that a little bit later. The Calvin cycle is happening in the stroma of the chloroplast, and the ATP and NADPH provide the energy to build the molecule. So again, light-dependent reactions are the power plant. They're producing the raw materials. Calvin cycle is the factory. We're using it to build. Okay, and I've used that analogy before in cellular respiration, but in reverse. So let's look at the light-dependent reactions real quick. So in the light-dependent reactions, when you have photons of light interacting with chlorophyll, the electrons in the magnesium atom become excited. And so this happens to all kinds of surfaces. You can put your hand in your car hood and it's in the summertime and it's hot because of the same idea. You're stimulating the electrons into a higher energy level, and when they fall back to the energy level they're supposed to be in, they give off heat or they give off energy. Okay, so if you are exciting the electrons of chlorophyll, one thing we don't want to happen if you're a plant is for that electron to fall back down to where it's supposed to be and give off heat. You want that to be captured into an electron carrier and taken somewhere so it can do work. All right, so we have our photosystems, and there are two photosystems embedded in the membrane, the thylakoid membrane, and they have reaction centers which have chloroplasts, chlorophyll embedded into the proteins. So here we have on the right a photosystem. And notice these little arrows. When the photon of light hits a photosystem, photosystem 2 is going to be absorbing 680 nanometers of wavelength of energy. Photosystem 1, 700 nanometers of energy. Wavelength. The photon of light is going to excite the magnesium. The magnesium is going to be liberated. And in fact, it actually bounces around from chlorophyll to chlorophyll until it gets to the reaction center where you have what's called the primary electron acceptor, which grabs the electrons at their highest energy level. So here they're low energy and they're building and building and building, and when they get here, they're high enough energy that the reaction center can grab them, and then those electrons are going to be used to do something. What are they there to do? Well, because the electrons are leaving at their higher energy level, we can't have magnesium atoms losing electrons because, of course, that's going to be a problem. Eventually, they'll run out. So water, which is part of our equation, provides replacement electrons for the chlorophyll. And there's a special thing that the plants do called photolysis, which is splitting of water. And this creates 4 protons and 1 molecule of oxygen for every 2 water molecules. Okay, so from that, we have oxygen being released, and that's our waste product. Okay, so, and on the right here is just a nice picture of the membrane, the protein that the chlorophyll is embedded in, and here are all the chlorophyll molecules embedded in the membrane. So this is the thylakoid space, this is the stroma, this is the thylakoid membrane. All right, then what we have, which is opposite of cellular respiration, is chemiosmosis. We're doing our electron transport chain early. So we've got our electron transport chain happening, and whoops. Here we have our photosystems, and so I'm going to walk through this briefly. So we started off with our photons of light hitting the first photosystem, the chlorophyll. It bounces around, it gets grabbed by the primary acceptor, and then it gets shuttled through oxidation reduction atoms to a shuttle molecule called plastiquinone. Plastiquinone transfers the electron to a cytochrome complex, very similar in the mitochondria, and you know what happens then. Protons are pumped into the thylakoid space. Now, when this happens, we're building up a proton motive force inside the thylakoid space. So low protons on the outside, high protons on the inside. Could we not do light and do a pH change and have this happen? Definitely. So all we need is protons building up in the thylakoid space. If we follow the electron through its path, it gets grabbed again by another electron carrier to the second photosystem. Now let's pause it there and talk about the photosystem 1. Photosystem 1 is being stimulated by a different wavelength of energy, 700 nanometers. Same thing happens. The electrons get grabbed by the primary electron acceptor and they leave. But nice thing is, the electrons that are coming from photosystem 2 just replace those that are lost. We don't have to do photolysis again, which is what's happening here, to replace the electrons. We already have them to be replaced coming from the second photosystem. So if we follow the electron some more, there's another electron carrier called the ferredoxin complex, which grabs the electrons and takes them to a very important enzyme attached to the thylakoid called NADP plus reductase. And the electrons are being added to protons to make NADPH. NADPH goes to the Calvin cycle. Now if we come back to our protons that are in the thylakoid space, because we're building up a proton motive force, because the membrane is impermeable to protons because they're charged particles, there's only one place for the protons to go. So again, the analogy I used in class was like a dam, and there's only one place for the water in a dam to go, and that's out the pipe at the bottom of the dam. In this case, that pipe is analogous to ATP synthase. So the protons move through synthase, we make our ATP, and the ATP gets taken to the Calvin cycle. So these are all the light-dependent reactions. And a lot going on here, and I would be happy to go back over them in class. What do you need to know? You do sort of need to know how this works and what the products are of the light-dependent reactions. What are the reactants? Light is coming in. We've got our chlorophyll reacting with the light. We've got our water that's donating electrons. What are our products? Oxygen, NADPH, and ATP. And this is just showing you the chemiosmosis, very similar to what's happening in the mitochondria. Okay, and we're going to stop here, and I will talk about the flow of electrons at some other point. So I hope this helped a little bit. If I went too fast or if you're confused, please post questions on Edmodo before Monday or write your questions down, and I'll be happy to answer them in class on Wednesday after our lab. See you later.